So I think we can start now. So as I mentioned to you, Janice, so the introduction today is in Spanish. And after that, we are going to start with your talk. So well, I'm going to switch to Spanish. So bueno, buenos días por estar conectados con nosotros en el programa de Frontiers in Genomics. Y para mí es un gusto presentar a la doctora Janice Cook. Janice es profesora de genómica de bosques en el Departamento de Ciencias Biológicas en la Universidad de Alberta, en Canadá. Su carrera la inició en la Universidad de Victoria y después, por un tiempo, estuvo trabajando en el Weinhauser Technology Center en Estados Unidos. Y una vez que trabajó ahí, hizo su doctorado en la Universidad de Alberta. Después hizo un postdoctorado en la Universidad de Florida y también fue investigadora asociado en la Universidad Naval. Después de eso regresó a la Universidad de Alberta en el 2005. Es una experta en genómica, fisiología y bioquímica de bosques y la respuesta que tienen estos a patógenos. Tiene más de 70 publicaciones y tiene una contribución muy importante en la relación de los pinos o bosques de coníferas con organismos parásitos de estos. Ella es corresponsable de lo que se llama el TRIA4, que es un proyecto internacional del genoma de bosques que financia el gobierno de Canadá. Es la directora en jefe del Tree Physiology Journal y coordina la División 2 de la Unión Internacional de Organizaciones que hacen investigaciones en bosques. Recibió recientemente el C.D. Nelson Award de la Canadian Society of Plant Biology y también recientemente recibió el premio Killman and McCullough por enseñanza de excelencia en la Universidad de Alberta. Bueno, con eso termino. Y with that, Janice, thank you very much again to be here with us. Now we're ready to hear your talk, and I'm sure we are going to enjoy it.
Well, thank you very much, Mario, for the kind introduction. I have to apologize that my Spanish is not great, but my French helped me understand a little bit of the introduction. It was very gracious. Thank you. I want to thank everybody for taking time out of their day to join us today. It's really a great pleasure and a great honor to be with you all today. And I especially thank the students who were with us yesterday and join us again today. We had an excellent session yesterday. I was so impressed with the questions and the discussion that we had. So congratulations to the students for really coming from such a great program to be accepted into a great program, coming from that program and really showcasing what they've learned in those program courses and through those experiences and their questions yesterday. So I'm going to share my screen now. All right, and I'm just going to move a couple of windows around. I think we should be good. So are we good, Mario? Is everything looking okay? Excellent. So today I'm going to talk to you about a system that we've been working on in my lab for a little over a dozen years now. This is the mountain pine beetle system. And one of the reasons why I chose to talk about this work to you today is because mountain pine beetle has also been an issue in Mexico. And so some of you might be familiar with the damage that mountain pine beetle has caused in the landscape and what happens post mountain pine beetle. So oftentimes we acknowledge our colleagues and our collaborators and our students at the end of our talk, but I really want to acknowledge all of the hard work that these folks in my lab have done over the years on this project. This project marries ecology, it marries genomics, it marries biochemistry, it marries physiology, and so all of these students at some point have been out of their comfort zone to learn new techniques, whether it's being in the lab or in the field or both. I really also need to thank our many collaborators who help make this research possible. I need to thank our funding agencies, and particularly my colleagues on our newly funded project, TRIA4, that enables us to continue this research for the next 5 years. So I'm going to start off with a little bit of an introduction to the mountain pine beetle system, and then I'm going to give a talk that's in essentially 2 parts. In the first part, I'm going to talk about how we've used genomics in a physiological and biochemical sense to look at the interactions of the host with mountain pine beetle and the fungi that they carry and how that's affected by the environment. And then I'm going to switch gears a little bit and talk about some of our recent work that we've been carrying out with quantitative and population genomics. This has been an amazing collaboration with a group of geneticists to start connecting genotype with phenotype, because I believe very strongly that this is the grand challenge that we face in contemporary biology, and I think genomics really is the key to do this. And then I'll end with a few perspectives. So here is our beast, Dendroctonus ponderosae. It's about the size of a grain of rice. And this is a bark beetle that attacks mature pines, burrowing through the bark to get to the cambial zone between the bark and the wood, where they excavate these galleries. And through these galleries, then these mated females will lay eggs. Those eggs become larvae. The larvae then will overwinter. Here's one little guy up here that escaped when we peeled this log. And the next spring, then they complete their development and they will escape their host, who is now well on the way to mortality, to find a new host to infect and ultimately kill. And so these beetles then can reach really high population levels. And so on the landscape, we see the devastating consequences of these outbreaks, where large swaths of pine are killed in a rather dramatic and sudden fashion. You see on the ground all of these red dead trees that eventually lose their needles. And these trees then become really, really flammable because they have these dead, tinder dry branches. The needles themselves that follow the ground are very dry, very flammable, and they're also filled with pitch that was produced in the attempt of these trees to defend themselves against these beetles. So the beetles don't act alone. They carry with them a very complex microbiota. The students and I talked about this a little bit yesterday, that this is an area that is absolutely ripe for metagenomics. And amongst the actors that they carry this group of ophiostomatoid fungi that we call the blue stain or the sap stain fungi. The most prevalent of these is a character called Grossmannia clavigerra. And these fungi then are carried by the mountain pine beetle as spores just behind the headpiece, which looks a little bit like a Darth Vader mask. And they will grow into the wood, into the sapwood or the actively conducting wood. And they cause some pretty extreme changes to the morphology of that wood. And ultimately, they will cause the tree to seal up the regions where this fungus is growing into the sapwood, and that blocks water transport. And this is why those trees are turning red, the needles are turning red. It's not because the beetles are attacking the needles, it's because water transport to those needles has been interrupted. And because of that, then those needles essentially dry out. So since 1999, when this outbreak was first detected in a historic area where we've seen mountain pine beetle in the past, we have seen more than 27 million hectares of pine forests in Western North America, from the Yukon all the way through south to Mexico. My apologies for not having Mexico on this map, have been killed by mountain pine beetle. And so this outbreak has really expanded. The reason why we have so much devastation is it's expanded from this historic zone that stretched from south central British Columbia and Canada through several of the US states and into the very northern part of Mexico. That was the historic zone. But we saw populations grow to some extreme levels, partly because of forest management practices and partly because of some unfortunate collision of dry summers and mild winter temperatures, allowing these beetles to reach huge populations, which then saw them expand into novel habitats. And these novel habitats were spreading north, they were spreading to higher elevations, and they were spreading eastward across the Rockies. And so in this region, you'll see that, in fact, they moved rather rapidly through this zone of hybridization between lodgepole pine, which is a host that has a long history of coevolution with mountain pine beetle, across this zone of hybridization to a new host. And this new host is jack pine. And jack pine is a boreal forest species. It hybridizes with lodgepole pine, and jack pine then actually has a range that goes all the way to our Atlantic provinces in eastern Canada. And so this was a huge cause for concern. So we actually documented that host shift from lodgepole into a new host, jack pine, for trees that were successfully attacked in 2010. And this paper was a game changer. And it was a game changer because of the policy implications. There were very many scientists who said, we knew this was going to happen. And then, yeah, there were other scientists who had said, well, you know, it could maybe happen in a lab, but it's not going to happen in real life. And it did. And so the policy implication was that this was no longer a regional problem in one part of Canada, but now became a national problem. And so it triggered a number of policy decisions in provinces east of Alberta, where this hybrid zone exists. And it triggered a number of policy decisions at the federal level. So this paper really opened this idea that we had a native species, but it had actually become invasive in a new species. And because of that move to a new species, changed the scope of what forests were under threat. So upon that research, we really started to focus our questions on how is mountain pine beetle going to do in these novel environments? And specifically what we were interested in is how are interactions with the host pines going to alter the beetle's success? Are population dynamics going to be the same as they are here in the historic range, or are they going to be different because of factors at play in these novel habitats? And so one of the really important things that we had to keep in mind is that we were tracking natural spread, but there was also a risk of accidental introduction into points east of Alberta. And one thing that we know from other experiences with invasive species in Canada is that transport of firewood by tourists and campers is actually a prime way by which invasive forest insect pests get transported into new regions of the country. And so partly because of the 2011 paper that we published, there has been a national initiative to try to educate people on the importance of not transporting firewood. But despite that, sometimes you get cases like this where people are in fact selling mountain pine beetle infected firewood in infected regions and People can actually just take it anywhere because it's not tagged, so it can't be tracked. And we have other cases like here in Jasper National Park, which is in the Rockies, where trees that had been infected with mountain pine beetle were cut down, but people were taking these logs before they could be taken away because they represented a free source of firewood. So we have to be aware that natural spread can happen, but we can also have human-mediated spread. So mountain pine beetle attack density really matters, and it matters to the success of the beetle. At low attack densities, mountain pine beetle can really only overcome the defenses of stressed and weakened trees. Whereas at high attack densities, they can pretty much take down any tree. And they're actually going to seek out the most healthy, the most robust trees because they will support the maximum brood. And so what we have here is a function of mountain pine beetle success as a function of attack density, where at low attack densities, normally in a well-defended tree, those beetles are not going to do so well. As attack density goes up, they're going to do excellent, and we're going to see that of the brood that is laid by mated females, that most of that brood is going to survive. And as that attack density gets super high, they start to compete for resources and those brood numbers go down. But where it matters is at the leading edge of an outbreak. And so here our attack densities haven't reached maximum levels. They're actually relatively low as those pioneering beetles disperse and try to find new hosts. And this is where a well-defended tree is going to be able to fend off those attacks, whereas an undefended tree will not. And so it's our belief that the trajectory of this curve is affected by the genetics of the host and the environment in which the host finds itself. So we've spent a lot of time over the last 15 years or so comparing the responses of lodgepole and jack pine to try to get at the question of whether jack pine presents, in the view of a mountain pine beetle, a different host. So we know that lodgepole pine is like chocolate cake. Mountain pine beetle just loves it. And so the question, when mountain pine beetle moved into jack pine, is whether or not mountain pine beetle would find it to be an equally suitable host, a better host, or not as good a host. So was it going to be chocolate cake, or was it going to be Brussels sprouts? And so we've been trying to understand that question using molecular and genomic tools. So one of the things that comes into play is the environment in which these trees thrive. So lodgepole pine and jack pine actually occupy different ecological niches. And so because of that, where their ranges overlap and they form this hybrid zone, this hybrid zone is actually quite complex. It's a mosaic hybrid zone where we find not only every shade of integration between these two sister species, but we'll also find patches of these pure species interspersed in smaller and larger clumps within this larger hybrid zone, or this zone of integration. And this is really reflective of the differences in adaptive traits that these two sister species have. And so I'm not going to show a lot of our data today, but jack pine is more adapted to drier environments, sandier soil, poorer soil. And so we see jack pine present at lower elevations in drier regions of that zone of hybridization, whereas lodgepole pine is found at the higher elevations. It's found in richer soils. It's found in regions where annual precipitation is higher. So this gives us some clues that the physiology of these two species has to be different. And so we have actually looked at a lot of the traits related to, for example, coping with drought and water limitation. And why that matters is here on the eastern side of the Rockies, we're in this continental climate, and in many years we experience drought conditions. And actually this year, we are experiencing drought conditions in some parts of the province that are equal to or even more water deficit conditions than what was experienced in the driest years of the 1930s. So here we see this map plotting the precipitation through our growing season, which starts about mid-May. And you can see that very many regions of the province have extremely low precipitation through the growing season this year. And so from ecological studies, we know that those stress trees tend to be favored by mountain pine beetle when they're at lower densities, because mountain pine beetle can overcome their defenses. And so we wanted to be able to get at the molecular reasons why that might be the case. So to look at that and to compare our different species, we've done a lot of studies with seedlings in growth rooms, and we've also done some field studies with mature trees. And I'm going to focus mostly on our seedling studies, but I am going to talk a little bit about some of our field studies as well. So the thing about mountain pine beetle attacks or attacks with the fungi that they carry, so if you artificially inoculate these trees, is that the tree will try to contain that invasion through developing what we call lesions. Now, these lesions aren't exactly like a hypersensitive response. So for example, a lot of these cells in here are living, but we can use that then as a measure of the tree's reaction to having been invaded. So we often measure lesion length as a first measure of the tree's defense response. So what we see is that lodgepole pine produces longer lesions more quickly than jack pine does, and that water deficit prolongs that period of lesion development relative to the well-watered condition in both of these species. And so, so we've interpreted this to mean that under well-watered conditions, we get this rapid lesion development, particularly in our co-evolved host. As time passes, we get continued cell division in that cambial zone, and we get new sapwood that's laid down, which actually will cover up that lesion when we still get active growth. That doesn't happen in these trees that are under water deficit condition. They actually slow their growth and eventually stop growth relatively early in the growing season. And so we see that same pattern in lodgepole and jack pine, but the lesions in jack pine are much smaller. And we see these same sort of patterns in mature trees where lodgepole pine produces much larger lesions than our jack pine. And these lesions are greater under water deficit, or sorry, under well-watered conditions than they are under water deficit conditions. So we see these massive anatomical changes, as I mentioned earlier, and I'm just showing lodgepole pine here for simplicity, where we get these distortions of our parenchyma cells, so our metabolic workhorses within our wood and our bark, and you can see deposition of starch grains and phenolic positive compounds in here, and you see really massive changes to the wood. When we look a little bit closer, we see actually within these tracheids, these water conducting cells, that we have all of these tyloses. And so even though the fungus hasn't grown into these cells yet, we are getting blockage or cavitation of these tracheids to prevent the movement of the fungus through these tracheids. So it's a proactive measure by the tree to try to contain the spread of the fungus that either was inoculated into these trees or carried by the beetle and through that process inoculating the trees. What we see, though, is that these tyloses are much more numerous in lodgepole pine than they are in jack pine. And so this is manifested in how much of that conducting wood is blocked. And that translates into how much water movement is blocked up the tree. So in lodgepole pine, with inoculation, we see a great decrease in hydraulic conductivity, or the amount of water that can move up that stem. And that's really illustrated here with these dye experiments, where in the inoculated lodgepole pine, we see a lot of blockage of tracheids that cannot carry that dye. In jack pine, we see a minimal effect of inoculation on our hydraulic conductivity, despite the fact that we see blockage of those tracheids. So we see compensation by jack pine, and this is consistent with the idea that jack pine is better adapted to low water availability conditions. So this gives us a first hint that jack pine has ways of dealing with attack that may lead to less mortality and may be able to overcome those attacks a little bit better than lodgepole pine, which is more susceptible to not being able to transport that water upon those attacks. Another thing that we see that's different between jack pine and lodgepole pine is that jack pine, upon inoculation under well-watered conditions, produces a lot of these what we call traumatic resin ducts. So these are resin ducts that form in newly produced wood in response to attack, and they're going to produce all of that pitch that we see coming out of those holes that the beetle bores into the tree as it's trying to attack, so those pitch tubes. So jack pine produces a lot of these traumatic resin ducts, compared to lodgepole pine, which really doesn't mount much of a defense of its wood compared to jack pine. It's producing all of those blockages in an attempt to contain, but it's not producing a lot of defensive compounds to make those differences. Under water deficit conditions, though, jack pine is not producing nearly as many of these traumatic resin ducts. So this would be a way in which jack pine defenses would be compromised by water deficit conditions, even if it has those compensatory mechanisms for continuing water transport, even in the face of being attacked. So we also wanted to look at the monoterpenes that we find in those resin ducts. And so we looked at the bark monoterpenes in this case, because it's the bark that the beetle is boring through as it first attacks. So this is the first line of defense that the tree has against this invader. So here we have lodgepole pine and jack pine under well-watered and water deficit conditions. We have control, wounded, and inoculated. Sorry? Everything's okay?
Okay.
Everything is okay.
Great. Okay. So there are a lot of monoterpenes that are produced by mountain pine beetle, and we can look at them collectively as the total monoterpenes. And I'm also looking at a few cases where we've got really stark patterns of individual monoterpenes. So I'm going to just walk you through this because there's a lot of data here. Well, what we see under inoculated conditions is an increase, not only in total bark monoterpenes, but several of these monoterpenes that are important in the communication between the beetle and the tree, and important for the beetle's success in the tree because it uses these compounds in production of pheromones, for example. So we see this increase upon inoculation with the fungus. But what we also see is under water deficit conditions, an increase in total monoterpenes, as well as several of these individual monoterpenes. And so we take a look at these data and we interpret it to mean that the tree has increased its constitutive defenses with this stress to better protect it against other stressors as it's dealing with this water deficit. But what we see, which could be important, particularly in seasons like this, where the tree is trying to protect itself against incoming beetles, is that when those trees are inoculated that are under water deficit, they don't see that increase in monoterpene levels. Rather, they see a decrease, an attenuation in their ability to synthesize new monoterpenes. And so our interpretation of this is that they've actually invested so much of their resources into producing these preformed defenses under conditions, in fact, where their photosynthesis is limited because of stomatoclosure, that they actually don't have the resources available to produce these de novo monoterpenes upon attack. So these are our lodgepole pine. We see different patterns in jack pine where we don't see the same strong responses. We also see differences like alpha pinene produced at much higher levels in jack pine, say, compared to lodgepole pine. We see less myrcene produced by jack pine compared to lodgepole pine. And so my colleagues that work on mountain pine beetle are looking at these mixtures to determine how they would affect that interaction with the beetle, and doing some very sophisticated experiments with flight, host detection, and other important parts of that interplay that happens between the host and the beetle as the beetle finds its host, attacks its host, and colonizes its host. So we went on to look at other genes that are involved in the defense response. We looked at chitinases, which are part of the protein defense arsenal. And we looked at a couple of genes, well, several genes actually, and I'm only showing two here, that are involved in synthesizing those monoterpenes. And we see a pattern of expression that supports this overall idea of increased constitutive expression of a subset of these genes under water deficit condition, and other sets of genes that show an attenuated induced expression. So we see less expression of these genes after attack under water deficit conditions. So we went on to do some transcriptomics. These data are a few years old now, and so they're actually done with microarrays. And so we see within our large set of differentially expressed genes that we have a subset of defense-associated sequences that supports this idea of an enhanced constitutive expression of defenses under water deficit, but this attenuation of the induction that would happen normally under attack. We also see some genes that show this delayed reaction, that they are upregulated under water deficit conditions when they're attacked, but that response happens later than it does with well-watered trees. What we're really interested in is identifying signaling components that might explain some of the differences in the responses that we see between jack pine and lodgepole pine. And there's a lot of detail on this slide, but just to say that we found lots of transcription factors, for example, that may contribute to the regulation of this defense response. A lot of these are known actors in the defense response in angiosperms, for example. So if we take a page from the angiosperm paradigm, what we saw was several genes that would indicate that jasmonate was involved in the defense response. So jasmonate biosynthesis genes, jasmonate signaling genes. We also saw genes that were involved in ethylene signaling. And so this would suggest to us that Grossmannia clavigira was a necrotrophic pathogen. This had been assumed, but there hadn't actually been any evidence of this. So we decided to look for jasmonate and salicylate to determine whether or not the signaling process could reveal whether we were dealing with a necrotroph or a biotroph. And so it turns out that we have a good increase in jasmonate, both in our lodgepole pine seedlings and our jack pine seedlings under inoculated conditions. Water deficit doesn't affect this response. Even at 28 days, we still have some residual jasmonate. But what we see is not much of a response of salicylate except for under water deficit conditions and mainly at this really late time point. And so we're actually not sure how to interpret this big increase in salicylate that's only under water deficit conditions and really only under jack pine. But we feel like it's probably not part of the actual defense response, but related to some other process that's being affected by the impact of the inoculation process and the invasion process by the fungi. So this provides us with some good evidence that jasmonate is in fact the signaling compound that's invoked when we inoculate with Grossmannia clavigira. And so we can assume that Grossmannia clavigira is a necrotroph. We have that evidence now. What's intriguing in the last few years is labs that work on Arabidopsis and other annuals have pointed to abscisic acid as maybe a hormone that we know is involved in stress response in our angiosperms, but maybe it's also involved in crosstalk in mediating responses in response to pathogen and pest attack. And so we wanted to look at abscisic acid levels because we know in our angiosperms, especially in leaves, that we get this big increase in abscisic acid in response to water deficit, but it hadn't really been looked at in conifers. And it certainly hadn't been looked at in stems. So we did look in lodgepole and jack pine for abscisic acid and some of the metabolites of abscisic acid. And we do in fact see a large increase in abscisic acid, particularly at our later time points in our water deficit seedlings. And these differences between treatments are actually not significant. So we see large increases in abscisic acid, but no effect of the pathogen inoculation. So it is possible, in fact, that abscisic acid is mediating this crosstalk between our water deficit response and our defense response. Now, ethylene we hadn't looked at in seedlings since we didn't have that assay working at the time that we looked at JA and SA, but our collaborator now has that assay working. And so we actually set about looking at abscisic acid or at ethylene levels, jasmonate levels, and salicylate levels in mature trees that were either inoculated with Grossmannia clavigira or naturally attacked with mountain pine beetle. And then to make sure that they only get attacked by mountain pine beetle, we've put this fine wire mesh over the trees once they were confirmed as attacked. And here's one of those trees here where the adults are attacking. And so what we do see is, again, a large increase in jasmonate under Grossmannia clavigira inoculation. We see an even bigger increase in response to mountain pine beetle natural attack. But when we look at ethylene, we only see that increase in ethylene in the Grossmannia clavigira attacked or inoculated trees and not in the beetle attacked trees. So what this tells us is that ethylene is in fact a part of the signaling mechanism of the trees in response to this necrotrophic pathogen, Grossmannia clavigerA, it isn't part of the defense response against this herbivore, mountain pine beetle, and the fungi that it's carrying are clearly not a major factor in the initial response, initial defense response of the tree to attack. So although the fungus is important for the growth of mountain pine beetle because it's able to invade that sapwood, acquire nutrients, block that water transport to create this nice dry environment for these beetles to develop. In fact, it isn't part of the initial defense phase. It may be part of a later defense phase when the beetle is laying its eggs and there's a need for a depressed defense response in the region of these eggs and developing larvae. So we've just started some RNA-seq on these mature tree experiments where we have the Grossmannia clavigerA attack and the natural beetle attack to compare in more detail these responses to the beetle as an herbivore pest and the necrotrophic pathogen. So hopefully we can dissect out more of the downstream differences between these two marauders. So now I'm going to switch gears and talk about our first attempts to start linking genotype to phenotype. So if we walk on the landscape after a major mountain pine beetle outbreak, you see a lot of red dead trees. But in amongst these trees, occasionally you find a survivor. And so in this stand, which happens to be very close to where I grew up in my hometown, almost all the trees that you see in this picture are on this side of the road are pines, and almost all these trees are dead, but you have a couple of survivors. And so the question is, are these trees just lucky? Did they not get attacked because they were small, they're inconspicuous, they just escaped by random chance alone? Or is there something to these trees genetically that enabled them to escape attack because they're not as good a host? So they have poor host quality, meaning that maybe they're well defended, or maybe they didn't produce the chemicals that draw the beetle in, or maybe they resisted attack because the beetles were unable to colonize. Could have been many reasons why these trees would not be appropriate hosts. But is there a genetic component to that host quality, or are they just lucky? That was the question that we asked almost 10 years ago now. And we weren't really even sure what we would find because of the technology that we had available. But it turns out with some of our new tools from some of our collaborators, we could start addressing this question. So the first thing that we needed was a population to test. And my ecologist colleagues in British Columbia actually produced a population that we could test. And this population was created by walking in woods like this, collecting cones off of our lodgepole pine survivors and victims. And this is possible because lodgepole pine keeps its cones on its branches. They retain the cones, and the cones stay closed because they're serotinous. So they require heat to open because lodgepole pine is a pioneering species, part of primary succession. This enables these cones to open, say, during forest fires. The seed can spread, and the lodgepole pine can regenerate. So these cones are kept on these trees. You can see a few of them if you look very carefully at this picture. And our colleagues then collected cones off of dead trees and off of trees that were still green. And off of the dead trees, they collected these cones from prior to the outbreak and after the outbreak. So the cones that were collected prior to the outbreak probably were produced, well, clearly produced by the dead mother, but probably also had a father who also would have died in the outbreak because so many of the trees had been attacked. So we expect that both mom and dad here have a host quality that made them attractive to the beetle during the outbreak, whereas the survivors, we collected cones that would have been produced after the outbreak, so recently produced cones. So both parents had to have been survivors. Those seeds were collected, the seedlings were growing up, and then we collected needles for DNA analysis. So I should mention that we collected from several sites. I should say my collaborators collected from several sites across 4 different localities in this region of central British Columbia, where the outbreak was really fierce in the early 2000s. So this was a level of epidemic populations where very few survivors actually made it through. So we analyzed these. We did genotyping using Affymetrix chip. This was provided by Sally Aiken's group at UBC. This is a Affymetrix Axiom chip. And there are 50,000 SNPs on this chip. We were able to validate almost 15,000 of these as high quality SNPs that were poly high resolution. And we did this in almost 500 individuals, and then applying a number of different techniques. So we did genomic selection. We also did some GWAS. We've also done some outlier analyses to find the genes that predict survivorship, find those genes that show allelic differences between survivors and the victims. And then we took out the genes that showed genotype environment correlations because we didn't want to include any potential false positives that had made it into the positive bin because of geographic location or because of environmental correlations. So this was a triage step. And so through this analysis, particularly combining genomic selection and GWAS, we found a selection of about 128 genes that predict survivorship. And amongst these genes, there's some very interesting annotations for genes that actually have a small chance of being involved causally in this, this ability of these trees to evade attack. And so that will be our next step, will be to look at these genes in more detail to see whether or not they're just linked to loci that predict survivorship, whether in fact they are involved in some aspect of host quality. And the other side of that will be the physiological and the biochemical analyses to look at the progeny of these survivors and victims to determine what physiological and biochemical traits might differ between these two groups of trees. So just ending on a few perspectives, we're more than 2 decades into this epidemic, but mountain pine beetle is still an issue. Despite the fact that more than 27 million hectares of pine trees have undergone significant mortality to date, we still have active outbreaks in Alberta. Those outbreaks continue to build and continue to threaten the boreal forest. We see that water deficit does, in fact, affect a tree's ability to defend itself. And it likely does that because of this idea of the trees with their conservative, long-lived lifestyle, enhancing constitutive defenses, but not having the resources available to mount an induced response. So lodgepole and jack pine have a lot of similarities in their responses, but they also have some intriguing differences that might actually render jack pine to be not as good a host, despite the fact that lodgepole pine is the co-evolved host and therefore has had the opportunity to mount specialized defenses. But through circumstances, that naive host has traits that seem to lend it better protection, at least against the necrotrophic fungus. The other part of host matters is that we have discovered signatures that can distinguish lodgepole pine victims and survivors. And so the next step then is validating those genes that predict resilience and doing the functional validation of a subset of those genes. And so with that, again, I want to return to thanking the members of my lab, both past and present, for all of the hard work that they've done on this project, particularly the 6 fabulous folks that you see here, as well as our great collaborators with whom we would not be able to do this project. And with that, I thank you for your attention, and I'm open to questions.
Thank you very much, Janice. So now the very nice, very interesting talk. So the seminar is open for questions. So you can raise your hands or just, well, Yvonne Rosenstein.
I unsilence me and show my face too. You've looked at all the genomics and genetics of all those 2 pines, but have you looked at differences in the genes that are expressed by the beetles that colonize one pine or the other?
You know, that is an excellent question. And one of the challenges that we've had is getting those samples to the people who do those analyses, because they have up until now being in Vancouver, and so getting the beetles to them has been a bit of a challenge. They've really concentrated on looking at the beetle responses just in lodgepole pine, but our new collaborator is definitely going to be looking at those beetle responses through RNA-seq, and what he's particularly interested in is looking at how the beetle faces winter. So we know that cold temperatures will kill the beetle. And we know the colder that is, the more beetles we kill. But it's not an on-off switch. It's more like a dimmer switch. The colder it gets, the more beetles we kill, but we never kill them all. So how is that cold affecting the beetles that survive? I mean, that's really a major question. And so our collaborator is going to be looking at that question and looking at it within the context of the historic hosts from central BC, lodgepole pine from more northern areas that we know we can genetically distinguish, as well as the jack pine. And so stay tuned. He's going to be able to give a fabulous talk on this in a couple of years.
All right. Thank you so much.
So the next question is by Arnaud. Arnaud? Arnaud?
Yeah, there he is.
Arnaud?
Yes, I have a question about, do you think that gymnosperms have systemic response? Because all of these, attacks from pathogens look like if they are not able to spread the signals that one tree is attacked and the rest is not responding.
It's really a good question, Arno. And in fact, we have looked, inside and outside of the lesion to see whether the molecular response was the same or different inside of and outside of that lesion. So we haven't looked, 10 meters up the tree, but we've looked, maybe a few centimeters outside or 15 centimeters outside of those lesions. And we do see that kind of systemic response. And part of that is that these trees really rely on that pitch production, the resin production. If you think about anything that happens with a pine tree or conifers in general, their response is just to produce a lot of pitch and hope that they push out the marauder, and they hope that the toxic compounds in that pitch will block and kill the marauder. So that pitch is not just produced within that lesion. It turns out that it's produced in cells that are actually quite a bit lower, as well as in the lesion, and then they're transported into that lesion area. So we do know that there is a systemic response, but what we have not been able to figure out is whether it works through the same classic mechanism as what we've shown in poplar, for example, where we know that that defense response really relies on phloem transport, xylem transport, and that there's a very specific path that systemic response moves through, and it's very quick. We haven't been able to track that as well in our conifers. So what we see is that there is a systemic response, but what we don't know is whether it is as efficient, because conifers really do tend to try to compartmentalize, and so maybe it's not.
And another naive question, if I may.
Yes.
Do you look at the side of the predators, like I would imagine woodpeckers, or I don't know?
Yeah, you know, we had a fabulous discussion about woodpeckers yesterday. They are actually we use them like our Twitter. So if we are going out to the woods and we're trying to locate our site with the beetle attacks, we listen for the woodpeckers, and then we just follow them. We use the GPS too, but we follow the sound of the woodpeckers, and they will lead us to the site. So they are really efficient at finding other bark beetles, including mountain pine beetle, and they are super efficient at stripping the bark off and getting to them. Flickers will do the same thing. What we also know is that there are competitor beetles out there, particularly in the jack pine, that like ips that will are more co-evolved with jack pine, and it seems that this provides some sort of competition where the even when beetles fly into an area, they may not have as many available trees to attack because they're sensing those trees have already been attacked. But where the ips is not necessarily killing the tree, mountain pine beetle would.
Thank you.
You're welcome.
So we have questions in the chat. So do you want me to read it for you, Janice, or you read it?
I can do that.
Okay.
Okay. So we have a question from AS, and I'm going to just read it out. Do you think that the microbiota of the beetle can be modified so that they do not die, but cannot infect pine trees so strongly? How much can climate change awareness help reduce beetle outbreaks? So yeah, so there are groups that are working on the existing microbiota of mountain pine beetle, so working on the gross maniclavidra to perhaps engineer them to deliver compounds to mountain pine beetle that would kill mountain pine beetle, for example. That work has been going on for a few years now, and that could be a potential way to reduce populations. There are others that had thought to produce lethality in these fungi, that maybe that would reduce the competitiveness of mountain pine beetle. That didn't necessarily work. And then there are others who think maybe introducing some new microbiota could potentially reduce the success of mountain pine beetle. So it's certainly something that is being looked at. There has been metagenomics done on mountain pine beetle. We don't work in that area, but there are some really excellent researchers that do. And certainly climate change awareness is vital. You know, the case of mountain pine beetle gets called up a lot when we talk about the effects of climate change on our forests. We know that mild winters definitely contribute to sustaining populations and also allowing them to grow to higher than predicted levels. We know that these hot, dry summers, like the one that we're having right now, the fires that are happening in British Columbia are a consequence. They are affecting those populations as well. So certainly it's top of mind when we think about rehabilitating the forests that we've lost to mountain pine beetle. What can we do to increase the resiliency of these trees on the landscape? What can we do to produce a forest or encourage growth of a forest that's more diverse to be able to handle these stressors? And the other thing that is being done is thinking about moving genotypes. So, you know, trees don't move, they don't pick up and move, but there's gene flow. And so there are groups that are looking at accelerating that gene flow, maybe moving more southern varieties more north because they feel like they have a better chance of being adapted to the environment that will exist there in 100 years. Because we always have to think about the long game when it comes to these conifers. It takes many, many, many years to produce a tree that is of merchantable volume, so something that could be cut down for timber. And I do believe very strongly, just as with the local food movement, where we want to get our food locally, I also believe in getting our other resources locally. So we want to be thinking about that, but we also want to be thinking about resilient forests for all of the other ecosystem services that they provide, like water provisioning and protection for habitat and that sort of thing. So those conversations are really, really important. They are going on. And we do a lot of public outreach to try to bring that message to the public, not only for those that have a connection to the forest, a strong connection, but also the general public that lives in cities that maybe don't think about these things every day, but when they're made aware of them, they're very passionate about them.
Thank you so much.
You're welcome. Patricia, you've had your hand up for a really long time, so I'm going to turn to you next and then maybe go back to the chat questions.
Hi, nice talk. And
Thank you.
I have two questions. And one is the population, the survival population is a really nice thing to learn a lot about this. But I wonder if, in addition to see, what you told us, have you also checked at what level these survivors are surviving? I mean, are they infected and then they have like any response, or they are not infected at all? So you don't have any trace that the beetles went in, or at what level, if you have seen it, these survivors are?
So that's a really excellent question, Patricia. So in these forests that had really high attack, the survivors mostly either had very limited attacks or no attacks. But what we do see in forests that maybe had maybe moderate level of attack, we see trees that actually had quite a few attacks, but successfully overcame them. And so we call these strip attacks, because what we can see if we were to take tree rings from these trees, is we see that they were attacked, in the 80s, for example, during the last outbreak. They survived that attack. You can see where the galleries were. You can see where that lesion was, and then they just continued to grow. But they just formed this wound tissue around the area where that attack was, and were able to survive it. So we actually see both kinds, but it depends on, at least in our experience so far, it depends on what the level of attack was. And I think it would really also depend on how systematic you were to look for the two types of survivors. And we haven't had the opportunity to look really super systematically over a large area, but I think that would be really, really important. So those are the two pieces that we've seen, but I think there's more to the story.
And a related question is that whether you are also checking the isoprenoids produced by the survivals or not, because, and that's why I wanted to know at what level was the surviving. And related to this, just briefly, these monoterpenes that you are looking at, are they coming from the MEP pathway, or are they coming from the mevalonic pathway, if you know?
Yeah, so these would be excellent questions from my longtime collaborator, Jörg Bullman. They actually did look at the monoterpene profiles of victims and survivors. They're a different set than we genotyped, unfortunately, but they did look at them, and there are fingerprints that can distinguish the victims and the survivors. But they were little trees. So now what we want to do, these trees now are, they're bigger. They are about 10 years old now, which, is about 6 feet tall in Canada. It's not very big, but still now we have at least a little bit more wood that's behaving more like wood and not just as a seedling stage. So it is actually on our books next season to collect materials for those monoterpene analyses and to pair it with some actual analyses of sap flow to see whether that makes a difference. So whether these are part of the MEP pathway or whether they're part of the MEP independent pathway, there is kind of evidence for both. We know that in Jörg Bullman's lab, there's good evidence for the players of the MEP pathway. But there are some reports that there could be an MEP independent pathway as well. But that's always the hard one to prove, right? Because what enzymes are you looking for? So, so certainly there is MEP pathway is implicated in the conventional monoterpene profiles.
Thank you very much.
You're welcome. All right. Mario, should we return back to the chat for questions?
Yes, the next one is Esperanza Martinez. Can you read it or I read it for you?
I'll go ahead. So Esperanza asks, does the fungi produce a purple pigment? It produces melanin, actually. This is thought to be protective to the fungus. I'll tell you what it is it's miserable for extracting DNA from these fungi because it interferes with the extraction process. So that melanin then is growing as the hyphae grow into the wood. The melanin pigment is being accumulated in the older hyphae, and that's where we get that blue stain color from. Okay. Our next question comes from a YouTube viewer, Joseph Dubrovsky. Does this beetle attack roots, and do roots respond somehow to the attack? Do the roots help under drought to survive? So the answer is they don't attack roots, but our sneaking suspicion is that the roots matter, and they matter a great deal, particularly for conifer responses to water deficit. So one of the other projects that I've worked on over the years has been with a longtime collaborator, Janice Cook, on mycorrhizal roles of mycorrhizae in water uptake and the roles of aquaporins produced both by the mycorrhizae and by the roots. And so we know that those mycorrhizal associations are really, really important to the water scavenging ability of the fine roots of conifers. And this is really important in conditions where the trees are under stress. So particularly in saline environments. So we have, in Alberta, we have these things called the oil sands, and the tailings ponds have a very high salt environment. And so for remediation for conifers, they have trouble actually getting water from those saline environments. We know that those mycorrhizal relationships are really important to helping the trees scavenge for that water. So those mycorrhizal relationships that the mature pines have are also important for any succession that happens after mountain pine beetle outbreaks. And that's work that another colleague of mine is working on campus. So the roots are important. The other thing about the roots that are important is that we know that they are an important reservoir of carbohydrate that's produced through photosynthesis. Those carbohydrates are shunted down to the roots and they're stored as starch. So in these drought years, we know that there is a large amount of translocation of these photostimulates to the roots and that the tree will draw from those for the next season. So that below ground woody growth is an important part of the tree's overall strategy to survive into another year under abiotic stress and also to combat biotic stress. So I'm not sure if that got at Joseph's questions.
I think so. It's okay. Then the next question is Tamara Lopez.
Sorry.
The next question.
Oh, there we go. Yeah. So because environmental stress has had an effect on the defense capacity of pines, could the spread of beetles towards the northern pines have been driven by climate change over the last decades? Yeah, so we actually believe that that's true, and we believe that it's true for a couple of different reasons. One would be those mild overwintering temperatures that we've had over several years of the outbreak, particularly in the early 90s, that allowed populations to grow really fast and to hyperdepidemic levels. When populations get really high, it turns out that beetle behavior changes. And so rather than just leaving their host and flying more or less horizontally to find a new host, they actually change their behavior and fly upwards. And so when they fly upwards, that allows for air currents to carry them longer distances. So we had a really massive long distance flight that actually introduced beetles into the northern part of the province in 2006. And that was critical to the establishment of that population in northern Alberta. And that wouldn't have happened without the populations in northern British Columbia growing to really, really high levels, which we believe was attributed to some of those milder temperatures. We also think that these beetles can persist because of those overwintering temperatures not reaching those really wicked temperatures every single year. So most years we have killing temperatures that will decrease those populations 90%, 95%, even 98%. But that's not enough to bring those populations down. That's just enough to create, well, we're all familiar with R values now because of COVID. Our R values with mountain pine beetle can be atrociously high. So if we can get down to 2, we're happy. If we get to 2 with COVID, we're not happy. So we believe that that's allowed for that population to rise more and also have those long distance flights that can carry them into those northern environments. The only saving grace has been that we've had some extreme forest fires up there at the time that mountain pine beetle was active around the 60th parallel, and it actually burned the forests where mountain pine beetle had invaded. So as far as we know, with the surveys that have been done, mountain pine beetle at those northern latitudes, north of 60, the numbers are really small and maybe not even existing right now. So that would be the only good news out of climate change is the increased fire severity. All right. Does sugar and sap change in the dry season? That's an excellent question. And I would say that from Esperanza, that question comes. And our evidence says, yes, that the sugar profile changes and the amount of sugars change as well.
So There is another question from Alejandro Daniel Camacho. Alejandro?
Hi, thank you. Can you hear me?
Yes, thank you.
Congratulations for your talk. Your speech is just fantastic.
Thank you.
I'm a former student from Simon Fraser with Dr. Barden.
Are you really?
Long time ago.
Oh, that's so cool. I should give you all of my questions. You could answer them better than me.
Well, not at all, not at all. But well, it's great to hear your research about your research. One question is that since you have the genetic signature of the survivological pines, is there any project to develop plantations for reforestation? And if that's the case, have you followed what's happened with those trees? And if that is, I'm pretty sure you are trying to do that.
Yeah, so that is really a great question. These data are pretty fresh. So we do have partners in the province of Alberta and the province of BC in their tree improvement programs. And so when you have one data point, it's not enough to make a tree improvement decision. So we need 2 data points. So our next step is to take these genomic prediction models and apply them to an independent population of trees. And so our collaborators in BC actually have collected cones and seed from victims and survivors in a different part of the province. And so we're going to test these models in the next couple of years on those independent populations. If we can still predict survivorship using our models, or maybe using an enhanced model, and we don't see that there is a growth penalty to these trees. So sometimes there's a growth defense trade-off, and that's something that we are going to be looking at. If there's a growth defense trade-off, then we would have to consider it a little bit more carefully. But if there is a limited growth defense trade-off, and we can use the validation models with our genomic selection models, then the intention is to add this to the traits that are being selected for in the BC program and in the Alberta program. And the province of BC is now using genomic selection in their tree improvement program. So they're well set up to take this up. The province of Alberta hasn't yet started using genomic selection, but they're very interested. So we're hoping that this would provide an opportunity for them. We have another project looking at a different pathogen of lodgepole pine, where we're actually further along in that process, looking to validate that model. So that's probably our proof of concept of whether the province would take up genomic selection as part of their tree improvement or not.
Thank you. Thank you very much.
You're welcome. Thanks for the great question.
So I think it was the last question. So I just have a simple question. Is just because I'm curious. So the genome of these pines is huge, right?
Yeah, it's massive.
Yes. And how many chromosomes they have?
20 to our best understanding.
Oh, 20. So you can do cytogenetics.
It has been done with pines. There are some groups in China that are doing these sort of cytogenetic analyses. So I have seen some, and so it would be a possibility for sure.
So that could help for the analysis of the genome somehow, for mapping genes or to correlate some regions or something like that. So I'm thinking since you don't have the genome yet.
Yeah.
So maybe some studies can some help. Cytogenetics always help. You can do cytohybridization and things like that in order to map some maps of regions.
Yeah, that's one approach that has been floated. The community has probably taken a little bit more of a classic approach, that they've produced physical maps from the same markers that are on these Affymetrix arrays. And there was a large project that was headed out of Spain where they actually did these physical mappings with the same markers in several species of pine. And the exciting thing is that the microsynteny between the species is really quite high, even between spruces and pines. So right now, the group in Vancouver for white spruce is using the physical map to try to snap on their longest contigs in ordered chromosomes. That's sort of the next step in the bioinformatics and using these physical maps. I think what's really going to help that process is if we can get to the next version of long read sequencing, because I think the long read sequencing is going to be, it's going to make a big difference with these, gigabase genomes. And especially if we can improve the fidelity of that sequencing. So that, and improve the bioinformatics of how to incorporate that with the physical maps, with the short reads. So that's really where we would like to go. But the cytogenetics is intriguing. And if there is ever going to be a breakthrough there, I believe it's our colleagues in China that will make it.
Okay. Well, thank you. Well, I don't think there are more questions. So thank you very, very much, Janice. It was a wonderful presentation. The history is very interesting. You have a very nice work. And we really appreciate your participation in the program.
Well, thank you very much for the invitation. It's been such a pleasure.
Hopefully next time we'll be in person.
That would be fabulous.
We hope that. Okay. So with that, we finish today. And thank you very much to everyone. And see you in the next seminar to all of you.